In today's activity, we are going to take a look at the process of transformation to create transgenic organisms. So I'm going to start over here. I'm just going to label this number one. So we're going to start with a person that does not suffer from diabetes. So that is going to be the disorder that we're going to be taking a look at for how this type of biotechnology, right, can make someone's life better. So we got our individual here, right? They do not suffer from diabetes. So we are going to extract the gene for insulin because that is the protein, right, that is needed in order to control for blood sugar, which is the main issue for individuals with diabetes. They're not able to either produce any insulin or enough insulin to control their blood sugar. So we've got our, we've identified our gene for insulin. And now what we're going to do is we are going to our human DNA, okay, and we are going to use a restriction enzyme. Right? Restriction enzymes are used to cut out the gene. Okay, so we've identified our gene right here for producing human insulin. Okay, so everything before that spot and everything after that spot, we don't want it. Okay, so we use our restriction enzyme, we cut that out. Okay, then we come down, okay, to our bacteria. Okay, so from our bacteria, their DNA, which is a plasmid, what we call the DNA for our bacteria. So the plasmid is removed from the bacterium and it is cut open, okay? And then the human gene is added in or is inserted, okay? So here is our bacteria cell, okay? We've taken out this plasmid, okay? And we have cut it, again, using that same restriction enzyme, okay? And we are gonna add in our gene, right, for human insulin. So now the bacterium, so the bacterium is now transgenic because it contains human DNA. Okay, because here's our human gene for insulin. So the recombinant DNA has been inserted back into the bacterium. Okay, so remember, it's recombinant DNA because it contains genes from both the bacteria and from the human. So that's why we say it's recombinant. Okay, so all right, this should be 8. That was 7. So next step, our transgenic bacterium reproduces and copies the human gene for insulin each time it divides. So we go from having 1 bacteria to 2, 2 to 4, 4 to 8, so on and so forth. And so our end product, right, is insulin. So insulin is produced on a mass scale, right, on a mass scale for people with diabetes, so for diabetics. So we've, at the beginning, right, we identified the gene for human insulin. We cut it out using a restriction enzyme. We found a bacterium. We cut open its DNA, which we call a plasmid. We inserted the human gene for insulin into the bacteria DNA or the bacteria plasmid. That bacteria is now transgenic and it contains recombinant DNA. Every time that bacterium divides, right, it also produces insulin, which can then be mass produced and sold to treat diabetes. So the reason we're using bacteria to produce human insulin is because bacteria reproduce asexually, and therefore their reproduction happens very fast, very quickly. So within a matter of hours, we have a very large supply, a very large quantity of insulin ready to go out and be sold for diabetics.